So today we're going to be talking about phloem transport and something known as the Münch pressure flow hypothesis, which is again another stellar hypothesis that when it was proposed, people thought it was crazy, but over time, as we had better and better experimental techniques, has come to be proven true. And this is going to be quite important because the last part of today, what I would like to focus on is plant biomechanics. And so we're going to go from water transport, nutrient transport, to biomechanics. And actually, this is all interrelated to each other. Okay, so phloem. In particular, what is phloem? Why should we care about phloem? So everybody should be able to identify kind of the key kind of structural parts of stems as well as roots, okay? But I want to remind you where the phloem is located, okay? In particular, the phloem is located near the outside then of the vascular transport network itself. So phloem transport. So remember, we have two vascular tissues, xylem and phloem. So far, we've mainly discussed xylem, but we're going to delve in right now into the phloem. So you should be able to tell us what the xylem is, the different cell types, the different aspects of this complex tissue. You should be able to identify xylem, but also you should be able to tell us its function, its purpose. Same thing with phloem. What is the purpose? What is the function then of phloem? The phloem is the food conducting component of the plant. And so it's the tissues then that making up this food conducting transport system, okay? Found in vascular plants. And so what do I mean by food? Food relates to the products of photosynthesis. And so sometimes we use the term photosynthate. Photosynthate is what is produced from photosynthesis. So remember, photosynthesis is producing sugars, right? And sugar derivatives. That's all photosynthate. And the phloem is going to be involved in transporting that product then of end result of photosynthesis. Okay. So phloem transport. So we talk about a term known as translocation. And so phloem transport is translocation. It's the transport of water-soluble products of photosynthesis over decent distances, long distances through the plant body. Translocation is the term. Phloem contains a sap-like quantity, a sap-like liquid, and it has high concentrations of sugars, mainly sucrose. It also has things like amino acids, right? But also organic phosphates. Oh my gosh, it's like almost an ideal food source for animals because we have the building blocks of proteins, we have sugar, we also have some nutrients inside. And so this is the forewarning. Plants don't like to share their phloem sap with animals, and because of which that's caused some tension and some co-evolutionary relationships. Phloem really moves at an impressive rate. So if you actually measure the velocity of fluid measuring going within the phloem itself, it goes on the order of about 10 to 100 centimeters an hour. Yeah, it's slow, but for a plant, that's pretty decent. Now, phloem transport, you should know based on the rate of transport, that's much faster than what's predicted from Fick's law. Fick's law is slow. Pure passive diffusion is slow. So how does a plant transport photosynthate at such high velocities, greater than what's expected from diffusion? The hypothesis that was actually proposed by Münch, a German plant physiologist, kind of in his heyday in 1927, was what's known as the pressure flow hypothesis, or the Münch pressure flow hypothesis. And it was an ingenious, really kind of crazy idea that eventually over time, this was kind of backed up then with a series of different experiments. And so what I'm showing you here are two different osmometers, all right? And so we've kind of used a few different osmometers diagrams anyways. It's great to see this actually in the lab in operation. But imagine two different flasks of water, okay? Pure water. And in those two different flasks, we're then going to connect then those flasks by one glass tubing going from osmometer B into osmometer A. But then we're going to have this kind of this bag kind of solution on the inside. And this is a semi-permeable membrane, okay? So we can get water moving in and out, but we can then concentrate. Think of these as independent cells, but this is a semi-permeable membrane. And each of those membranes then are in each of the osmometers. Those membranes are then connected themselves through another kind of glass piping. Okay. And so this is a physical demonstration then of actually what's happening. So imagine an osmometer A right here. In our semi-permeable membrane on the inside, so this is our fake cell, on the inside, we increase the concentrations then of solutes within that cell. So we naturally have a gradient in osmometer A from within the concentrated solution within this semi-permeable membrane, and then the outside of the membrane where we have close to pure water. Okay. So this high turgor pressure then in osmometer A is going to result in pushing water and solutes ultimately to osmometer B. Now, all that's happening here is the flow of water along a water potential gradient. So if you look at osmometer A and osmometer B, if you compare then the water surrounding each of these different semi-permeable membranes, you should be able to immediately tell something about what the water potential values should be, or the values of psi. So notice within our osmometer B, within the semi-permeable membrane, where we have a concentrated solution, high amounts of solutes. This could be sugars, it could be salts, okay? Some sort of ion, but we have a concentration there. And because of which, that's gonna change psi sub S, okay? So immediately there's gonna be a difference in psi sub S between the inside and the outside of osmometer A. And because of which, water then will move along a natural water potential gradient. Water will flow from the outside to the inside, basically creating a higher turgor pressure, okay? Basically increasing the water potential then inside of this. Effectively, this flow of water is going to act to push then the flow of liquid from the inside of the semi-permeable membrane to the other, okay? If anything I've said so far does not make sense at all, this is a chance to do a full stop and put in your notes. Please go back and review these fundamentals of water potential, because hopefully this should make sense that water should be moving from the outside of the osmometer to inside the concentration solution, because we have a concentration gradient difference and reflected in psi sub S. Now remember that because osmometer A and B are linked via this glass flask, right, through this little tube, water then is going to be moving from osmometer B to osmometer A. And the only mechanistic driver of this is this concentration gradient between the inside of our fake cell, the semi-permeable membrane, and the outside, and this concentration difference in solutes, okay? So high turgor pressure is going to be generated from this natural gradient, pushing then water from osmometer A to osmometer B. And this is then causing the pressure then in osmometer B to increase, and because of which we're going to get this movement of water kind of all around, okay? Now, here's another way that I can kind of show you this in a more kind of graphical sense, okay? It's actually great to see this animated, and I have some links, I believe, to some very nice animations for this as well. But what I would like to do is show you this, what looks like a weird kind of drawing, right? On the left-hand side, we actually have the xylem. So notice we have some xylem vessels or tracheids. And then right next door, we're actually gonna have some phloem cells, okay? So I'm gonna go back in my slides and point out right here, this cross-section of a stem. Remember, we have our living phloem, then we have our vascular cambium, and then immediately we have, as we go kind of right next door, we have the xylem, okay, the sapwood, okay? So remember, physically, xylem and phloem are right next to each other, and that's gonna be really important, okay? So I'm gonna go through several steps here, and I want you to please pay attention, because these steps are actually critically important in terms of how actually phloem works and how it actually transports, because it requires this tight coupling with the xylem. All right, so notice right here in this one cell, this is a crappy drawing, but this is a leaf cell, okay? And so this is your typical leaf. It's performing photosynthesis. It's making sugar or photosynthate from photosynthesis. That sugar then is going to be actively transported into the phloem. So just like our osmometer, like in osmometer A, we're increasing the concentration of sugars then within the cells, within these phloem cells. But remember, the xylem is right next door. And so remember, just because of water potential, the xylem transpiration stream is following a water potential gradient. But notice right next door to the phloem, there's another water potential gradient. And so this higher solute concentration is actually increasing the water pressure potential. And because of which, just through osmosis, water is going to be flowing from the xylem into the phloem. Now, not all the xylem water, just some of it, right? Because the phloem is not that big in the grand scheme of things, but some of that water is then going to start flowing into the phloem because it's following a natural water potential gradient that's set up within the plant itself. And so that flow then of water into then these phloem cells is going to create a high pressure environment. So the phloem is under positive pressure because of this action of increasing solute concentration, water actively then moving in under pure osmosis, creating high pressure environment. So the xylem is under negative water potential. The phloem is under a positive pressure potential. And because of which, we're actively pushing under positive flow water and everything else then that's within then the phloem itself downward. Now, the other key part of this mechanism is that we have two critical steps. Near the leaf, sugar is going to be actively transported into the phloem, but at some other part of the plant, say in the root or in the stem, we're going to be actively removing sugars out of the phloem, okay? And we call that active transport because ATP is required. So the plant is spending ATP to actively push sugar into the phloem, but in some points of the plant, some cells are going to be using that sugar to grow, to respire, to perform maintenance. And at those sites, we're going to be actively taking sugar out of the phloem into the cell. So ATP is going to be used. The other two key terms that I want to emphasize here is that we're going to have cells that are source cells of sugar, and sink cells for sugar. Source cells are where sugar is being produced from photosynthesis. Sink cells are where sugar then is going to be used to actively grow, to do respiration and kind of fuel and power, kind of all aspects of homeostasis and growth. Okay, sources and sink. So within the phloem, we have sources of sugar and sinks of sugar. And it's this active loading and unloading that creates then these concentration gradients of solutes. So notice near the sink, we have high solute concentration, because of which water is going to move from the xylem inside, creating high pressure. That high pressure is then going to push then fluid, because near the sink area, we're going to be actively unloading that sugar into the sink cells, creating a lower solute concentration and lower pressure. So then we set up this pressure flow so that fluid in the phloem is always going to be going from sinks to source, active high positive pressure. And notice water then is going to, at some point, as soon as the concentration gradient gets to a low enough point, it will move back into the xylem, basically recycling that water. This is an ingenious invention by Mother Nature. Natural selection has come up with this coupled parallel transport system that takes advantage of a natural water potential gradient, takes advantage of the fact that water will move by osmosis, okay, to high areas of solute concentrations to create this positive pressure flow, And then with active transport of loading and unloading sugars, we can then activate then the phloem. This is the pressure flow hypothesis. And it turns out that it works beautifully well. Okay. So let me show you a few additional kind of details. Okay, so these are the central steps in the Münch pressure flow hypothesis. The first step, at source cells, these mesophyll cells within the leaf, the plant is going to be actively loading sucrose into the phloem. This is an active process. It requires ATP to take sugar from near areas of chloroplasts, put it then into the phloem. 2, transport those sucrose into companion cells into the phloem. This then lowers then the water potential then within companion cells, and water then is going to flow via osmosis from the xylem into the phloem. The third step, the sucrose solution is then going to be forced into the sieve tube elements of the phloem, and the phloem then becomes pressurized. And this is then going to cause then the phloem to flow, okay, to actively move. But then the fourth step, a critical step, is that at the sink, so maybe in roots or in areas then of the plant that are actively growing, actively metabolism, the phloem solutes are then going to be unloaded. And at those sites, at the sinks, the water potential is going to become less negative. And this is going to cause then the water then to flow back into the xylem, basically being recycled. So the water potentials then of the source cells, okay, up by the leaves are not going to be affected because they're creating sugars as fast as they're going to be unloaded. So this happens really fast. We're going to be loading, 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 and somewhere in the plant, we're going to be unloading, 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 and this creates then this flow. Okay, so similarly within the sinks, okay, these are cells that are actively growing, respiring, converting sugar into stored compounds or starch, or using that to fuel the production then of new work. All right, so it's going to be used as quickly as it's unloaded. All right, now several good animations and more details into the pressure flow hypothesis. So please kind of check them out on your own time. Yes?
Could you maybe go back to number 3? There was a student that wanted to take a closer look. Okay, awesome, thank you.
So in number 3, okay, this is the third step. All right, in the third step, the sucrose solution then is going to be forced into the sieve tube elements of the phloem itself. So remember, the phloem is going to be consisting of these 2 different types of cells, these companion cells and these sieve cells. These are cells then within the phloem. And as soon then as the sucrose solution then is moved then into these sieve tube elements, the phloem then becomes pressurized under positive pressure. And this is then going to cause then the phloem then to move under positive pressure. Right? So this is really cool, because what this means is that as you go from the xylem, which is under negative water potential, it's being pulled. As you go into the phloem, it's actually pressurized because water is actively flowing into it, and because of which we're going to be pushing then that process of osmosis, and water then moving from the xylem into the phloem is going to push then the phloem along then this gradient going from the source to the sink. Okay. Ingenious. All right. So good animations, check it out in your own time. Now, how do we know that the pressure flow hypothesis is correct? Well, there's a number of different experiments that you can do. One thing that you can do is a series of puncture experiments. This is pretty, basically poke a hole in the tree and see what happens. So if you poke then a hole, like a glass tube, you put a small little glass tube or needle all the way into the xylem, what you're actually going to find is that if you then puncture the xylem, you puncture then that negative water potential, that suction. And what actually is going to happen is that air is going to be sucked into the xylem because it's under negative water potential. So you'll actually see air rushing into the xylem as it basically kind of forms this embolism and gets sucked up. But if you use the same needle and just puncture the phloem, what you'll actually find is that phloem sap, phloem fluid will kind of bleed out because it's under positive pressure. So when phloem is punctured, sap flows out, indicating that the fluid is pressurized. So when xylem is punctured, air is going to be sucked in, indicating that the fluid is under tension or under pull, negative pressure. Okay. So another kind of classic support for the phloem transport is something known as a girdling experiment. This is basically, how do you kill a tree or a plant? Well, the best way is to girdle, then basically remove the phloem, leaving the phloem and the xylem on either side, and basically watching what happens. And if you do that, because the phloem is located outside of the xylem, you're basically removing the phloem, but not the xylem. And this is called a girdling experiment. And over time, there's going to be this accumulation of solutes as it basically kind of gets dumped, and there's nowhere left to go here, right? And because of which, all of the cells in the sinks and the roots and everything are no longer receiving photosynthate. And because of which, those cells then that are below then this girdling point will actually slowly start to die. And if you want to kill a tree really quickly, this is a great way of doing this. Okay. And so these kind of early experiments provided evidence that assimilates or photosynthate were transported down the phloem, whereas fluid was moving then up and within the phloem itself. And so if you want a really fun time, if you can kind of wander the bowels of YouTube, you can find, people like this that like to tell you how, if you go out homesteading, and if you want to try to figure out how to best clear a forest for whatever you want to do with a cleared forest, they will teach you how to kill a tree. This is horrible. This is like botanical snuff films, right? So here he is, the heathen, the evilness. Look at this. He's girdled on the tree. He's gone in, right? He's gonna kill this tree. He's now gonna go through and he's gonna remove all of the phloem because the phloem is on the outside. Oh my gosh, ghastly. A horror film. Look at that. That poor tree doesn't stand a chance. So on the time frame of a few days, maybe a week or two, that tree will slowly start to die. I don't recommend going to the dark parts of the internet so you can find these horrible things, but a really cool experiment, and actually an experiment that we can use to study the details of phloem transport and then the inner physiology, is to actually use insects to help us out. And this is another evil experiment too. It's an insect snuff film. It's pretty bad. You can take these aphids. These are kind of the weird things. If you ever grow kind of like, kind of vegetables in your garden, and you go out and you want to try to see kind of how well your plants are doing, and you go out and you look at your plants and they're all stressed because they have these crazy aphids. Aphids are phloem feeders. These are insects then that specialize in going in and puncturing. They have this great little proboscis that punctures then the stem, and it basically feeds on the phloem, all the sugar. There's amino acids, remember, there's nutrients as well in there. And so these are horrible little parasites going in and actually feeding on the phloem. And sometimes you'll actually see a lot of ants associated then with aphids. And what's crazy is that these aphids, as soon as they kind of puncture the phloem, remember, this is like the phloem's under positive pressure, right? So it's like trying sometimes in a really healthy plant, it's like trying to get a sip of water in a fire hydrant, right? So as soon as the aphid then kind of punctures the phloem, because it's under positive pressure, you get this gush of phloem juice that kind of rushes then through the aphid, and the aphid has some specific adaptations to try to get rid of all that pressure. And in fact, here at the end of the aphid, out of the backside of the aphid, some of this phloem juice kind of comes out. And these ants actually specialize in kind of tending and harvesting aphid dew, basically phloem that shoots out under positive pressure through the aphid to the backside then of the aphid. And on really healthy plants, you'll get these ants then that specialize then on feeding then on phloem juice that comes out of the other side of aphids. I know, interesting, wonderful natural history, really cool evolutionary biology too. But another cool thing that we can do as plant physiologists is we can take advantage of the fact that these aphids, and horrible aphids, look at this, taking advantage of the hard-won photosynthesis of plants, where they poke their little stylet into the phloem, positive pressure, it all comes out. Well, what we can actually do is we can let the aphids do their thing, go in under a microscope, and we can remove the aphid but leave the stylet behind. Basically, chop off the rest of the aphid. And what's nice is that we have this nice little aphid size needle puncturing exactly into the phloem, and we can harvest phloem fluid in there, and we can study then at different parts along the plant how the phloem transport changes and how differences in sources and sinks then operate. It's really cool. And so we can analyze phloem sugar concentration to see if it actually changes with distance from the source and distance from the sink. And if you do that, what you actually find is it totally supports the Münch pressure flow hypothesis.